This is Intake Pest Management session. There's actually 3 folks going to speak. I'm Philip Roberts, Extension Entomologist, but we also have Dr. Jason Schmidt, who is a research entomologist, works in biocontrol, and Dr. Michael Toews, who works more on the IPM pest dynamics side. But we're going to start with Jason first, so carry on.
All right. Well, welcome to the afternoon. So thanks for coming, and thank you, Philip, for having me speak in this session with you. Philip asked me to talk about biological control, and in particular, how we might go about determining which of those predators out there are important at times and others not, and then how do we go ahead and maintain those services out in the field. So Biocontrol Services is a kind of a newer term for biocontrol, and that is those organisms out there or viruses that are against or provide service to pests that harm humans or their resources. So resources here would be cotton and fiber production. We can think of them then as the enemy of my enemy is my friend. That is, these predators and parasitoids and viruses and pathogens are our friends, and they will do a lot of the work for us if we can serve them. So to do that, we really need knowledge of the natural enemies that are out there, these enemies that are our friends. We have to conserve them in some way, and then we need to time other management tools around their activity and also use selective insecticides and pesticides when at all possible. These services are valued annually, globally at around $400 billion a year. In the United States alone, that equals around 4.5 billion. And I think we're on, this work was done back in 2006. I actually think we're on an upswing on this, and I would like to see this get a lot larger in the United States in the next few years, and I think that it will. This idea of biocontrol services and how and what they mean kind of starts like this. And once upon a time, it was about limited to this. That is, we have a certain set of pests. We know those pests have negative influences on our production, and in this case, cotton. We also know that there are predators out there and parasitoids, those enemies of our enemy, that exert negative effects on those pests and therefore provide a net positive effect towards cotton production. What's usually left out of this then is the producer. That is, the producer is also a part of this equation, and so is the consumer. And so, if pests are having a negative effect on that consumer and these predators are having a net positive effect, the net positive effect should help make decisions by the producer to either potentially have negative effects depending on this pest and the nature of dealing with that pest, or when possible have positive effects and benefit that control, which will circle back and help the producer. So then management practices can have negative effects or positive effects. So I'm just going to give an example, kind of a classic example of the negative effect that was even talked about on weeds just a moment ago. And that is, sometimes these chemistries can have unequal toxicity, or some pests are just better at dealing with chemicals than other organisms in the environment. So for insects, this looks a little bit something like this. So we treat a pest, and in this case you can see these are aphids, these are lacewing larvae. Okay, well, it looks like that has a significant effect on the lacewing larvae. However, it definitely did not take care of all of the aphids. And so the aphids come back again. However, we have very few of those natural enemies to deal with those aphids. And so we call that a pest resurgence. And in cases where the toxicity of this chemical to the pest is differs a lot from the toxicity on the predator, we have considerable amount of pest resurgence. That is, if this chemical is very toxic to the predator and only minorly toxic to the pest, that will definitely wipe out the predators, and we're going to have a really big problem with dealing with that pest in the future. So finding ways to combine the best types of pesticides that are very selective for the pest will help benefit those predators and have those predators do the work for us. So we can also facilitate biocontrol through a number of ways. I already mentioned the pesticide one, scouting, and what I always hear Philip say is get yourself a scout and a consultant to make sure that they're there, and using selective chemistries. So that would look if you look at that picture then, I changed the way this interaction diagram looks in the sense that this, by doing that, you lower the effect of pests, which increases the effect of these predators on that pest, and therefore that increases the increases the net positive effect of predators on production. So there are other ways to enhance this that are related, and that is insect diversity is commonly positively correlated with biocontrol services to a certain degree. So you see I drew this line up here. When we get up to certain degrees of diversity, some of the species tend to not exactly provide that additional services. So somewhere in here, increasing diversity is definitely positive to a certain degree. And there are ways to do that. A few ways are increasing the diversity of the landscape, increasing the types of things that are on the field margin, and you may have heard of things like beetle banks or ways to separate certain regions in a field to kind of compartmentalize different fields, which adds diversity to the overall landscape. By doing that then, this creates further natural enemies that can have that effect on pests, which lower the degree of that pest problem to a certain degree, and then therefore have a net positive of the natural enemies on production. However, and this is where I come in, however, this link here is not as commonly made. We talk a lot about these diversity effects and what they mean, but we don't talk a lot about that direct relationship between production. And a second thing that we don't often know a lot about is what these predators or what this diversity actually means. That is, we don't know what many of these predators out there are doing. And that's where I come in. So what we do know is that when we do things such as cover crops, through the last few years that I've been studying cotton here in Georgia, we add cover crops. What does it do? Cover crops not only, as Culpepper was just talking about, helps significantly with weed control, but cover crops provide habitat for animals to live, such as natural enemies. That habitat then allows them to grow their populations before you even have pest problems in the field. So, for instance, we've done this a few years now. The results tend to be pretty consistent. That is very early in the season through pre-emergence into squaring. Cover crops such as rye and clover definitely increase the populations and diversity of natural enemies in the field that can feed on things in early season like thrips. And that's particularly clear because early in the season, there's a lot less vegetation out there, so the natural enemies don't have any place to live. And the residue from these different cover crops allows them to have something to feed in. However, this later season one here is a little bit challenging because this material that was there earlier in the season, that residue is breaking down. And so we don't see much of an effect in terms of diversity and diversity and abundance. However, we do see effects on predation. And I've gone over already a little bit somehow because it's already at 13 minutes, which is pretty amazing. So what we do see affects later season, which is definitely a significant pest being these stink bugs. That is, we get increased predation on their eggs later in the season. So cover crops can have later season effects. And I'm going to swing through this. If it's all right, if somebody moderates me off, then I'm good. Okay. So here's the other way that I come in, and that is you see predators out there, or you see different insects out there, and sometimes we don't know their function. So what I do is I'm ripping those individuals out of the environment and trying to figure out what they do. And we call that molecular gut content analysis. That is, I grind them up, I turn them into mush, and I ask questions. And the question is, what are you eating? And so what we have in here, this is a food web, and I'm going to show you a few of them. Okay? What we have here is on this top area here, you see all these different colors. These different colors represent different taxa of predators. Okay? This blue one here is spiders. And I will tell you, in all the systems so far that I've studied down here, spiders are definitely providing or have major roles in pest management, that's for sure. Okay, so then they're connected down here to different pest groups that you would find in cotton. This is two-spotted spider mite, which is not a huge problem, it can be. Collembola, which is food, flies, aphids, so on and so forth. So if we take that information then and kind of try to organize it. So we take this is difficult to really see what it is, but it's cool looking and it's fun. This here allows us to ask questions now. And one of the questions is by taking all this data, that is, each of these, this kind of heat map of predation represents how frequently we found predator groups to feed on different things. So this is aphids, columbella, flies, thrips, spider mites, whiteflies, stink bugs, and ligas. And then these are all the predators. So they're ranked in this way. That means these taxa over here, these different predators, are eating a lot of those different pests in certain parts of the year. So I highlight those. And one of them, and you probably recognize this one. Does everyone recognize this one from out in the field? Yep, so this is a minute pirate bug. And this one is feeding on all sorts of things out there. So very beneficial. And a surprising one to me is this little columbellus. These are ground-dwelling things that recycle nutrients in the ground. They're using this as alternative prey. So alternative prey is good for keeping these alive in the field. So keep that face in mind. They're obviously not this big, and I would be frightened if I saw one that big. Another group that's really common in the fields are spiders, and one group in particular are lycosids. These are wolf spiders, and if you see one like this, if you see that right here, this is a pile of lycosids on her back. So she carries them around. This can be upwards of 300. If you see this on her back, don't squash her. She's going to disperse all those spiders all over, and they're going to be feeding heavily on all sorts of things. There's a few other groups that I see commonly in here. This is a jumping spider, and if you see web-building spiders between there, they're indirectly also contributing to biocontrol because they build their webs. And I'll tell you one example, that is when the whiteflies were going crazy in the fields, and sometimes they do that. If you look at that webbing, there are tons of whiteflies stuck in there. I do not have much predation on there at all, but indirectly they're contributing. Another group that's pretty diverse, and I want you to be able to recognize two larvae. You may have seen this larvae is kind of the more common ladybug larvae that you see, but this one down here you should also be able to recognize. This is skimnus, and it's a weird looking little larvae. It's smaller, and it looks like it has all these little projections coming out of it, but this is also a very good predator hanging out in there. And here's Philip Roberts' favorite, he told me, which is Geocaras. This is a very active predator in many of the systems that I've been in. There are two different species, and then the little nymph looks like these little gray things. Now again, this scale, these are tiny, these are very small. So you're not looking for something this large out there. Another group that's showing up really high out there, not too surprising, but these have three forms that you want to keep an eye on. The adult, which doesn't really feed on any of your pests. If you're seeing this in the field, this is really good. These little alligators, they ravage pests out there. So it's a good sign if you're seeing a lot of those crawling around. So to finish here, a few things that are unfortunate and are vulnerabilities in the current biocontrol system definitely have to do with whiteflies, as we have seen. Stink bugs, from my understanding of stink bugs, it's difficult, and their insecticides have to be used. I see parasitoids on them. Fire ants are contributing by eating their eggs, but in terms of me detecting them in the guts, I'm detecting very little predation using this technique on those. So to finish, let those biocontrol services work for you, and this is a matter of planning and trying to work with the environment to do a lot of the work for you. And with that, thank a whole bunch of folks, and thank you, and I totally went over time, and I'm sorry, you guys.
Anybody got a question for Dr. Schmidt while Mike takes up? You mentioned fire ants. Fire ants are also a very, very good predator. And then we have a lot of parasitoids. Mike, it's open at the bottom.
Come again?
It's open at the bottom. Click on that P at the bottom.
All right, so I'm going to try and recap some of the work that we've been doing on whiteflies in 2018. Thankfully, obviously 2018 was not nearly as severe as the pressure that we experienced in 2017. I'll hopefully be able to show you some of the factors that contribute to these populations, show you what we've learned and where we're headed with this. So no self-respecting biologist stands in front of a group of folks and doesn't talk a little bit about insect life cycles. Key take-homes I want you to remember on this is that, of course, we have an egg stage, and each individual female whitefly out there is leaving 150 to 200 eggs. And you think about the number of whiteflies that you see in a given crop, multiply it, and you can see that these populations can develop very, very rapidly on us. And that's really the battle that we're fighting, is how rapidly these things can increase when the conditions are all just right. Also, the generation time during warm Georgia summers can be as short as about 2 weeks. So in 2 weeks, you have that 150x increase, and that's really problematic for us, and you'll see how those increased. Georgia is blessed with great soils, nutrition, sunlight, rain, all these sorts of things that make it really palatable to grow great crops. And so this is the distribution of our cotton acreage down in the southern part of the state. But this is the overlap with our vegetable producing areas. And what I want to highlight with y'all is that the same whiteflies that get started in spring vegetables then move directly over to cotton. We produce whiteflies on 1.4 million acres of cotton, and then when we defoliate that, it's those same whiteflies, literally can be the same individuals, that are moving back into fall vegetables. And so this is going to be not just a one commodity type situation where we just need to control whiteflies in one commodity or another. It's critical that all agricultural producers buy into this idea that we've got to minimize our whitefly levels, and that's going to help everybody because it's the same whiteflies in each of those different commodities. This is the way we represented it in our cross-commodity extension bulletin that we put together. Literally all through the year, all 12 months, we've got some kind of an agricultural host that's a good host for whiteflies to develop and increase their populations. Of course, cotton is the largest acreage crop, but that doesn't mean that it's the most important, because it's just important that we have a host in the wintertime and in the early spring, and we've got to be mindful about these populations all the way around. In terms of the research that's been going on then, so this is a project that my lab has been working on for the past year. We have just over 100 traps, it was about 125 traps, 800 miles, and on a weekly basis we go out there and change each one of those. So here's an example of the traps. These are yellow sticky cards. That one's up in the air about 4.5 foot, and we've got a second one at each position that's about 6 inches off the ground. Give you an idea of the spread, so we're all the way from Vidalia, Georgia, up in our onion growing region, through Tifton in the middle, and then all the way to Dothan, Alabama. And so on a weekly basis, we're out changing those, looking at how this spread of whiteflies develops over the course of the year. Within all of those traps, we've got a number of different types of habitat, and we're going to be pulling that apart as we get farther and farther into the data analysis to get a better idea what crops or adjacent to what crops these populations are going to be first detected in, and then where they're developing over the course of the year. Of course, we're interested in cotton for our purposes, but as I'd mentioned, cotton is not the only crop out there that's a good whitefly host. So here's our overall curves. They're starting January on this end of the graph all the way to December on this end for all of 2018. And you see that our populations are very, very low in the early spring, the winter where we are right now. We get good freezing weather like we had overnight. That really allows us to drive that whitefly population down. Now the freezing events will kill the adults, but it doesn't necessarily kill the immatures. When they're tucked up underneath the leaves on cabbage and things like that, we've got to have repeated freezing events that are spaced out a week or two at a time to really drive that population lower and lower. You'll see that there's a slight increase in these populations that happened in about the month of April. And so one of the things that we discovered early on in this project is that it's not just silverleaf whitefly we're dealing with. That's a little critter called the Dewberry Whitefly. The adults looked identical to silverleaf. The immatures very different. I'll show you pictures there. The important part of the curve then is that mid-July, things really, really took off. And you can see where I've superimposed some of the different places where we grow those crops. And so you see toward the end of the cotton cycle, certainly soybean, if you're a fall vegetable producer, putting out tomato, pepper, snap bean, this is really, really difficult to deal with. Part of the reason that you have all this dispersal of whiteflies at that point is because we're chemically defoliating cotton. When the leaves fall off the plants, those whiteflies have got to go somewhere. So across the entire area, remember this is 125 traps, we're literally averaging 40 adults per trap at that point in time. This is where the hurricane hits, and you see a very, very fast reduction. We can't really plan on a hurricane every year, and in fact, hurricanes really drive whitefly populations very low. It pretty much kills every adult that's out there. I don't have data from a year when we don't have a hurricane, but my expectation would be that they would linger a little bit longer. In terms of splitting up the abundance based on area, if you think about Tifton as being right here in the middle, this blue area corresponds with the tallest peak in there, and then this red area corresponds with the second tallest peak. So the county extension agents that have been here for a number of years have been telling me, hey, look, it's Tift, Berry, and Colquitt counties, and that lines up very closely with what our traps were showing us, that this is kind of the epicenter right here in the middle around Tifton and Colquitt counties, Tift and Colquitt counties. And then the more outlying areas, let's say clear up to Vidalia and Dothan, were at a level that would be, a third of that or so. However, there were whiteflies across our entire study zone, and that's an important take-home, that it's not that they're just whiteflies right, in this part of the state. We had them spread abundantly across that entire area. Another take-home that we saw was that we expected the predominant number of the whiteflies to be silverleaf, which is exactly what we saw. I'm going to say 98, 99% was silverleaf whitefly. But there were some other things out there. There were at least 5 different species, and sometimes it's really difficult to separate. So you look at this one and say, well, that looks pretty similar to that one. You have to go find the immature stages. So there's the immature, the silverleaf. This would be the Dewberry Whitefly. See that very dark coloration, and you start looking for some different features that go along with the immatures. We had several different species of banded wings. See that this one is about twice as long as that one is. And so we're still kind of feeling our way through getting confirmed species IDs on those. Some of the genetic material is in gene bank, some of it's not. So we've got to work on, we just catch adults on the cards, so then we have to go back to that area and find an immature to do the actual taxonomy. This is some more information on that Dewberry Whitefly that showed up in April. That population really got large in just a few key areas. And when we got to look, and it was heavily influenced by the presence of dewberry, which is like a wild blackberry. And for practical purposes, if you think of it as the bramble or the blackberry that grows in ditch banks, that's what that thing is there. So there's the immature stages. You see that the immatures are actually black instead of clear or a little bit yellowish like what we're accustomed to seeing. Once we were able to start separating, okay, that's dewberry, that's not silverleaf, then we were able to regraph what was happening. And so April didn't look quite as scary after we realized that this species does not reproduce on cotton and vegetables. One other thing I want to point out is that the approach that we're taking is not just where are they occurring or where are they developing, but how do we manage these things. And so there's always insecticide trials going on. A very interesting area that we're pursuing right now is this naturally occurring fungus called Isaria. Isaria is common in the tropical regions of the world, kind of the Caribbean basin. We've had hurricanes the last 2 years in the fall, and we've blown up spores that came out of, let's say, Costa Rica, Puerto Rico, areas like that. And those spores land on the adults. They can land on immatures as well. You get this sporulation. It actually kills the whiteflies. We think that that's pushing our populations lower once we've inoculated in there. So the hope is, if we continue to see whitefly populations, that this particular fungus is something that's going to be endemic to our area. It's going to set up shop, and hopefully this is going to help us keep those whitefly populations lower. Right now it's naturally occurring, but we only see it very, very late in the year. We've isolated that. This is the wild isolate from here in Georgia, and we're looking at some bioassays, and as you can tell from that, about 90% of the lab colony we were able to kill in 7 days with a very brief exposure to that particular entomopathogen. So I think this is an exciting area for us. If you compare it, for example, to the kudzu bug, which was really, really a serious population issue for us 5-6 years ago. We had a naturally occurring entomopathogen that came in, a little teeny tiny wasp, and they have driven that kudzu bug population down to below economically important levels. None of us would have guessed that 5-6 years ago. So we're hopeful that there's some opportunities for that kind of stuff, but we're certainly just chasing it at this point. What I need to point out then is that yes, we're following populations, we're seeing spread, we're learning a lot more about how they develop, but that doesn't take away from the basic things that we've been preaching for a long time in terms of how do you manage whiteflies in cotton. These factors are still very, very important, and starting with things like winter weather. When we have really cold winters, when we get below freezing, that drives those populations lower so that they don't get started early in the spring. Smooth versus hairy cotton, smooth versus hairy leaves on your cotton makes a big difference. Hairy is much more prone to whitefly outbreaks. early versus late planting dates. And I know a number of you have seen this kind of stuff before. I just want to reinforce that this is all very definitely in play. And our job here is to figure out how to manage these populations over the long haul so that we suppress them and keep them below an economically important level. You know, things like irrigation. If you've got good rainfall or good irrigation, the cotton tends to do a lot better. When we had a year like we did in 2018, we did fairly well. But I think all of you would agree we're not likely to get that much rainfall again in the coming years. All right, so to kind of summarize, our populations are very predominantly silverleaf whitefly. Those populations, at least in 2018, really took off in July. Up through July, we didn't see a lot going on. We peaked in early September, and the population was overall truncated by a hurricane this year. We may or may not be able to plan on that in the future. Populations started earlier and built to higher levels in the Tift, Cockwood, Berrien area, but that's not the only place where populations existed. They were across that entire extent of South Georgia. And so all of our growers need to be aware of whitefly and how to manage it. And then finally, that local vegetation really influenced those captures. So we're learning a lot more about where do we want to put a sentinel trap, for example, to indicate, hey, whitefly populations will be building up earlier in the year. So that's something that we'll be asking additional questions as we keep pulling those data apart. And then the last thing I want to mention is that we've had great buy-in from a number of different groups to help us fund this kind of research. So the College of Ag, the University of Georgia, Georgia Cotton Commission, the Georgia Farm Bureau, the Fruit and Vegetable Groups, Soybean Commission, Cotton Incorporated, even the Georgia Legislature has appropriated money to allow us to really focus on different ways that we can manage these populations. And that enables us to make great strides in terms of what we can extend based on science. And then some institutional cooperation with Fort Valley as well as the USDA here in town, and a number of different departments. So a very good example of a multidisciplinary project. I'm going to stop right there. I will be around afterwards. I'm going to give Philip as much time as I can for him to go, and we'll take questions when we get all done.
Anybody got a question for Mike while I'm just loading up? All right, quiet group. Jay, you wait back there. All right, I'm Philip Roberts, Extension Entomologist, and I'm going to take a few minutes and just wrap this up. One of the things I want to say to begin with, being successful in any business is about making good decisions. Agreed? The only way we can make good decisions about insects and insect pest management is to know what's in the field. Hire a scout, hire a consultant, make the time to scout your crop yourself, but you have to know what's in the field. I do want to mention we do have scout schools each year. In this year, Tifton will be on June the 10th. And we also have a school in Midville on June 18th. So we actually cover scouting, cotton, peanuts, as well as soybeans. It's the same insect pest, we just count them a little differently depending on which crop system we are. Now in Georgia we have a good thing in terms of insect management. If we go back to the 1980s, prior to boll weevil eradication, prior to planting BT cotton, we sprayed cotton on average 15 plus times. Individual growers may have fields sprayed 25 times. And a lot of our growers today never grew cotton during that time. But even today, we're spraying cotton, somewhere between 2 and 3 times. A few fields a little more, a few fields a little less, but on average about 2 to 3 sprays. So that's a good thing. We actually have a competitive advantage over many parts of the country in terms of control costs for insects. And that's because you as growers do a lot of things right. Now I'm going to spend a few minutes talking about BT cotton. And what this slide is illustrating is actually the genealogy or the evolution of BT cottons we've been planting since they were first commercialized back in 1996. And what industry has done, they've added a second gene, and in recent years, a third gene. They did that for 2 reasons: to increase efficacy against target pests, but also for resistance management purposes. Now, what I want to share with you today is changes we're seeing in susceptibility of corn earworm to these technologies. Now, one of the things, if we just look at BT cottons we plant, we're basically planting cottons with 4 different BT genes, if you paid attention. And what I'm illustrating here is actually those same 4 BT genes are present in both corn as well as cotton. All right, and that's an important point for us to understand. Again, if you just look, the very first gene that was put into cotton back in 1996 was Cry1AC. But it's available in corn, it's available in cotton. The second gene in Bollgard 2, or twin link, it's also in corn, you see? Cry1F, that's the second gene in wide strike, it's in corn, it's in cotton. It's in the wide strike. And then the third gene, the dip gene. But the same 4 genes are in corn as in cotton, and that's an important point. And the reason it's important, when we look at the biology of corn earworms, what are corn earworms doing right now? Corn earworms are in the ground in an overwintering stage, okay? And what will happen is we begin to warm, these corn earworms will complete a generation primarily on wild host. Sometimes we'll see them on seedling corn, but the bulk of this first generation of corn earworm that emerges from overwintering survives on wild host plants such as clovers. All right, there's some wild geraniums, there's a lot of different broadleaf weeds that corn earworm can feed on. It takes about a month to complete a generation, especially once we get into the summertime. But the second generation we see usually occurs during the month of June, late May and June. That's on field corn. And that's an important point. And the reason it's important, a high percentage of all the corn earworms funnel through corn. There are not a lot of other wild hosts that corn earworm is on during this time. So there's a lot of selection pressure in terms of resistance or susceptibility, whatever you want to call it, that occurs in corn. For that reason, since there's not a lot of other natural hosts, corn growers are required to plant a 20% refuge of non-BT corn. If you're a corn grower, I encourage you to do that. Because when these corn earworms complete a generation in corn, that's when they come to cotton, which we typically see in early July, mid-July, 10th, 15th of July. All right? So during July and August and even into September, we'll have a couple generations of corn earworm on cotton. And I get the question, what about a refuge on cotton? We have a refuge for cotton, but it's something called a natural refuge. Back when we had single gene cotton, we had to plant a refuge, but with the current multiple gene technologies, we rely on a natural refuge. And what that is other crops such as soybeans, peanuts, grain sorghum, some annual summer weeds, but that's the refuge, okay? So there's 2 things going on here. It's important to understand that a lot of selection pressure occurs for resistance in corn. And what happens in corn is what we're going to see in cotton. Now, it's been a long time since we dealt with corn earworms in cotton. Just a couple of reminders. If we're going to have problems, it often starts in blooms. Okay? That's kind of the weak link. If we're going to see a problem, it's near that uppermost bloom in a cotton plant. If you have a corn earworm attain about 3 days of age, that's a 1/4 inch in length, he's very likely to survive and cause damage on that individual plant and maybe a neighboring plant. But again, so that's where we're going to be focusing a lot of our scouting efforts is on blooms, dried blooms, under bloom tags. And if we hit our threshold, which is about 8 larvae per 100 plants, we need to react promptly. So one of the things that we don't talk about much anymore is tobacco budworm. And even when I go to a professional meeting and I visit with my colleagues, we hardly ever mention tobacco budworm. But if you think back, some of you can remember back before we had BT cotton, tobacco budworm was terrible. It actually almost put Alabama out of the cotton business. And BT cotton kind of came in and saved us. But the reason we never talk about tobacco budworm anymore is all the BT cottons we plant still provide excellent control of tobacco budworm. They're still super sensitive to all of these BT genes. Now, what I want to focus the rest of our time on is talking about corn earworm, because changes in susceptibility to the BT toxins is occurring with corn earworm. And this is a rating chart. You can find this in the production guide. We have some text in there you can read. But the 3 gene BT toxins which are available still do a very good job controlling corn earworm. But if you look at the 2 gene offerings, not quite as good, but there are differences, particularly with wide stripe, not as active as twin wing or Bollgard 2. Okay? You just need to understand what each of these technologies brings so you can manage appropriately. Now, I'm up here talking about changes in susceptibility to BT cotton. And in reality, we haven't had any problems on BT cotton, any widespread problems. We spray a few fields here and there every year. But this is just a summary of some data from the last 2 years where we established a trial at Tifton, Plains, and Midville. And basically, we went at the end of the year, right before harvest, and we counted the number of bowls that had been fed on by corn earworm. And I did that for each technology. And even in the non-BT cotton, and this is 2 years of data from 3 locations, we were less than 0.5% of bowl damage. All right, the point I want to make here, whether they're susceptible or resistant, if they're not there, who knows? Just not a field problem. I've to show you some information that there is some changes in susceptibility. And it's my opinion, if we have populations, we're going to see a problem. Actually, if you look at this data a little differently, you can calculate percent control when actually the wide strike is the weakest of the 2 gene cottons. It actually only reduced bowl damage by 75%. Okay? So, but again, it just shows there are differences in the technologies, but that's representative in that rating chart. Understand what you got and manage appropriately. The other thing I'll mention, I forgot to mention, we don't see problems in corn in the field, in cotton, but we do in corn. If you look at a field of 2 gene BT corn this year, I feel pretty confident you're going to see corn earworms in the tips of those ears. 5 years ago, that corn would have been clean, so something's changed. So we do see biological changes in the field. Now, Dr. Greg Payne from West Georgia University, the Cotton Commission funds him, and then we do a lot of other things too, looking at susceptibility to these BT toxins. This is some data that actually began back in 1996, but what he's doing is he's looking at the concentration of a sprayable BT, it's called MVP2, but it basically works just like the first gene that was put into BT cotton. And what these bars represent is the dose required basically to kill corn earworms. In 1996, what is that? Probably 50 parts per million. Look where we are in 2018. It's at least 1,000. So something's changed. You see that? It's pretty telling. Should we be surprised it's changed? Probably not. That gene's been out there for 20 some odd years. So what I'm most concerned with is this is some other data where we're looking at a little different way of monitoring susceptibility. This is where you're looking at actually dried leaf tissues. But if you look at the first gene, we tested 8 populations this past year. 3 out of the 8, 38%, we termed resistant to the first gene. And what we think of as resistance as entomologists, if it's 10 times harder to kill, we tend to think of that as resistance. But of those 3 populations that were resistant, they were all at least 225-fold tougher to kill than our susceptible check. Okay? So they were highly resistant. Not every population, but it's out there. If we look at the second gene, for example, that's in Bollgard 2, and it probably behaves very similar to the second gene in twin link, half the populations were either 49 to 85-fold resistant. So there's some problems out there in the population. The third gene is still super susceptible. We did not test the second gene in wide stripe because it's not real active on cornea worm to begin with. We also look at susceptibility to pyrethroids. You know, we don't spray a lot of pyrethroids on any pest, on cornea worms in any crop. But we do have some concerns of how good we'll do based on what's happening in other parts of the country. For example, the state of Mississippi no longer recommends pyrethroids for cornea worm because they lost efficacy. We still think we have efficacy, but we just need to watch these and make sure we monitor behind sprays when we're targeting cornea worm. So kind of just to sum up, and then maybe we'll have some time for questions. One of the things I would encourage you to do is if you'll go to our website, ugacotton.com, myself and Mike and entomologists from across the U.S., we're in a group called the Cotton States Arthropod Management Working Group. But we developed a publication called Best Management Practices for Cornea Worm in U.S. Cotton. And I just want to hit a couple of these things real quickly. And again, this was written statewide. But the other thing I want to remind us of is we do a lot of things good in Georgia. Okay? We do a lot of things good in Georgia. We don't even realize it. But we had Dr. Schmidt talk to you about beneficial insects. We need to start paying more attention to beneficial insects. You know, we've drifted away from conservation as much as we should. But there's information such as conservation of beneficial insects. Actually, bollworm or corn earworm prefers hairy cottons. But we're not going to plant hairy cottons. Why? For silverleaf whitefly, right? But there's some information in there you can read. Lindsay, we don't want to use too much nitrogen. Why? 2 reasons. Green lush crop, insects like that. Green lush crop gets big and thick, hard to get insecticides down in the middle of the canopy. So a lot of little things. And again, we got to get back to management. So last slide. What I want you to take home from my talk today is you need to understand that susceptibility to these BT traits are changing. The third gene still looks real good. The first 2, it's changing. We haven't had a lot of problems having to spray BT cotton in Georgia, but they have in other parts of the country, particularly the Mid-South and North Carolina and Virginia. And again, I don't think we've had any problems because we just hadn't had the populations in the field to see it. The writing's on the wall when we look at our monitoring data in terms of susceptibility. And what it tells me, or what it should tell you as a grower, hey, I need to make sure I do everything right to manage risk and do everything we can so the problem doesn't occur. Now, one of the things I do in my job is I know it's bad in some other parts of the country, and I'll ask the question of why. And me and Jason and Mike will talk about why is something occurring. Why is it so much worse in the Mid-South versus Georgia? What are things that are different? And in my mind, there's 2 reasons. They have a lot more pressure, a lot more higher infestations from corn earworm. Number 1 is I like to think of the corn-cotton ratio. So if you think of how many ears of corn there are on an acre, what, 30,000 maybe, big ears. Then you got sucker ears, but just the more corn you have, the higher populations you have moving from corn to cotton acres. If you have higher corn acres than cotton acres, what's on cotton is going to be a lot higher than if you have a lot higher acres of cotton like we do in Georgia versus corn. So I think that's one of the reasons we have low populations, but I think the number 1 reason, and the reason we need to be most concerned about or try to practice, is that in both of those areas, they spray tarnished plant bugs. And I like to think of it as playing cards. They got dealt a bad hand. You know, in the Mid-South, they're spraying plant bugs, not because they want to, because they have to. Not just 1 time, not just 2 times, but 4, 5, 6, 7 times. They're spraying a lot of plant bugs starting when cotton squares. That's a critical time. And Jason's data showed it. You know, when we got to squaring, that's when the beneficials really started setting up shop. And the numbers are fixing to really increase. So the point I want to make to you is over the last couple of years, I've heard more and more discussion about plant bugs in the state of Georgia. They're an occasional pest. We scout for them. If we have a problem, we address it. But by no means do we need to be spraying plant bugs on an area-wide basis, because you're really knocking out beneficial insects at a critical time if we spray plant bugs. So I think that's one of the reasons, and that's my opinion, that they tend to have to deal with corn earworms much more than us. Okay. Any questions? Comments? I had told my other speakers we would have 0 attendees, but we had 10.
What is tarnished plant bug effects on cotton anyway? When we get to some level, what is that?
So pesticide credits, you know the drill. We're done, film man. Green is commercial, white is private. Tarnished plant bugs, when are they an issue? Tarnished plant bugs primarily are going to feed on small squares. If they feed on a square, it will be aborted by the plant. It will come off. It takes a few days. We have a threshold of 80% retention. And we also have sweep net thresholds as well as drop cloth switch thresholds, but it's all about retention. 80%. We want to hold 80% of the fruit. Any other comments?
If you wanted to plant a variety, just Beauregard 2,
Yep.
would you be concerned as a farmer?
So I was asked this exact question a few days ago. And this was a farmer. He says, well, should I do anything different? At this point in time, I would say no. You know, there's still some very high yielding Beauregard 2 varieties. There's some high yielding wide strike varieties. You need to understand what you got. I'd say this, if you plant a wide strike cotton, you better have a scout. You better be prepared to react. Beauregard 2, I think you better have a scout. I I understand not everybody has their cotton scouted, but if I had somebody looking at my cotton or I was scouting my cotton, I'm gonna plant a Beauregard 2 if it's the highest yielding cotton. Now, if I don't have a scout and I ain't got time to look at it and I want to manage risk, you may look at some 3-gene things, but I just don't think we've had the problem that we make any drastic change yet. Now, I would try to do all the other little things, but we're not to that point. Now, other parts of the country, that question would probably get a different answer, especially in areas where they can't spray a pyrethroid and they have to move to something like a Prevathon or a Besiege. You know, then we start getting pretty serious in terms of dollars. But right now, I don't think you do anything different. You understand what the technologies bring, and you just manage them. Okay? Hope y'all enjoyed today, and we'll see y'all next year.
Appreciate it.
What you think?
Good to see you.
Y'all got finished picking down there, or is it still?
What's that now?
Y'all finished picking in that neck of the woods?
we did.
Yeah, you know, my mother-in-law's got a little place down there.
Where does this information go? I think they videoed it last year too.
Yeah.
So where is this going?
I don't know actually. Entomologist is the company. We were hired privately by somebody I assume within the commission. So it's either going to be the commission's records, or if people weren't able to make it, then I can make copies and send out to those who ask. So that would be my assumption.
So it's video and you have sound that came in here, so sound should actually be pretty good.
Yeah.
Do you guys do any editing on it?
Absolutely. Well, actually, we're supposed to get copies of all the presentations so we can incorporate the actual slides.
Wayman was, did you know Wayman? You know Tommy?
I know Tommy, very good.
Wayman was.
Because I know
That's way years ago.
I guess I just got to contact them.
Okay, yeah.
So I'm looking right there on the
If I wanted
corner.
to use it.
Absolutely.
Yeah.
And
So you package them up as separate presentations?
I can actually get you my contact info.
Let me see if I got my card here. I did have some earlier.
We call